Good evening, all. Let us talk one more topic under biochemistry, that is structure of immunoglobulins. So, objectives of our presentation, we need to discuss regarding structure of immunoglobulin, which includes Y-shaped structure and variable and constant regions, antigen binding sites, and heavy and light chains. Coming to the structure of immunoglobulins, these are proteins, and these immunoglobulins as abbreviated as Ig. And here we can see in the picture light chain molecular weight 25 kilodalton, whereas heavy chain molecular weight is approximately 50 to 70 kilodalton. And in the picture we can see here there is variable region on heavy chain, variable region on light chain, and constant region on light chain and constant region on heavy chain. And these are connected by the disulfide bridge. So the same like here in detail you can notify the picture. So this is a picture of immunoglobulin. And the immunoglobulins have same basic structure and they consist of 4 polypeptide chains, that is 2H and 2L chains. H is indicating high and L is indicating light. The 4 chains are linked by disulfide bonds. And the pairs of heavy and light chains combines to form Y-shaped molecule. And here the heavy chains may be 5 types and are designated by, and here we can see the heavy chains are categorized or maybe 5 types. So these are designated by the Greek letters like alpha, gamma, delta, mu and epsilon. Here the immunoglobulins are named as their heavy chain type. For example, IgA, IgG, IgD, IgM and IgE. And the L and H chains are subdivided into variable and constant region. And here most H chains consist of 1 variable and 3 constant domains. So coming to the structure of the immunoglobulin, it is looking like Y structure. Immunoglobulins have a Y-shaped structure composed of 4 polypeptide chains. How many? There are 4 polypeptide chains and 2 identical heavy chains and 2 identical light chains. And the variable region of the both the light and heavy chains from antigen binding site. And the enzyme papain digestion splits the immunoglobulin molecule into 2 fragments named as FAB, fragment for antigen binding, and FC, that is like crystallizable fragment or fragment for complement binding. And whereas coming to the variable and constant regions, each immunoglobulin contain or the chain consists of variable V and constant C, that is denoting with the C region. The variable regions are highly diverse and are responsible for antigen recognition, while the constant regions determine antibody effector functions. And coming to the antigen binding sites, the tips of the Y-shaped structure. So this is the tips of the Y-shaped structure formed by the variable region of the heavy and light chains. So these are formed with the heavy and light chains and which contain antigen binding sites. And these sites are highly specific to particular antigens and allow antibodies to recognize and binding to them. And whereas coming to the heavy and light chains, the heavy and light chains are larger and more complex than light chains. So each heavy chain is linked to a light chain by disulfide bonds. So these are connected by the disulfide bonds forming a functional antibody unit. And let us summarize our topic. So far we have discussed regarding this structure of immunoglobulins. So these are resembling with the Ig immunoglobulin. And these are basically known as comes under the category of proteins, and basically all the immunoglobulin having the structure of Y shape and which consists of the 4 polypeptide chains, so and also which having variable and constant regions, and in this we having this antigen binding sites. So these will be binding towards the antibodies, and also which is having the two heavy and two light chains. I hope my students you have understood the concept of structure of immunoglobulin. Have a nice day. Once again we'll meet.